The methods of disruption of cells. This is one of the methods of downstream processing when the desirable fermentation product is intracellular. That means if the location of the product is inside the cells, then there will be the first step in downstream processing, that is disruption of cells. Whatever the methods we have studied so far, like filtration or centrifugation, which are useful for the removal of cells, in most of the cases the desirable fermentation product is extracellular. It is not always the case. Sometimes, for example, in case of intracellular enzymes like lipases. So lipases are produced intracellularly as well as extracellularly. So in such cases, the first step in order to liberate lipase enzyme from inside the cell to outside, the first step will be lysis of the cells or disruption of the cells. So for the intracellular products, the microbial cells must be lysed in order to liberate the products, and then there will be purification of the desirable fermentation product. There are several methods of cell disruption. The first method is physicochemical methods of cell disruption. It involves lipid shear. Shear means breaking with liquid, solid shear, agitation with abrasives, freezing and thawing, and ultrafiltration. These are the methods of physico-chemical nature. Another set of methods is chemical methods. It makes use of detergents for the lysis of cells, osmotic shock, alkali treatment, and treatment with enzymes, cell wall degrading enzymes. Let us see these methods in detail. So the first one is physico-mechanical methods. As the name indicates, there will be use of physical shock and mechanical shock in order to cause the lysis, or in order to cause the breakdown of cell membrane, breakdown of cell wall, in order to form the pores or holes in the cell membrane and cell wall, so that the intracellular contents will be liberated out. So first in that is cutting with liquid. So this liquid generates a force, and that force is responsible for the lysis of cells, and that is why it is called as liquid shear, cutting with liquid. So for liquid shear, for the lysis of cells, an equipment which is used for this kind of mechanism or method is high pressure homogenizer. So as the name indicates, these homogenizers works under pressure. So there is application of pressure and homogenization, and that is responsible. So this is homogenization of liquids which contain microbial cells. So whatever the force which is generated in the homogenizer, that is liquid shear, will be responsible for the lysis of cells. This is a typical diagram of this high pressure homogenizer. You can see the parts of this, that part A, as it is labeled, it is the pressure transducer, so which generates the pressure. B part is control system that controls the pressure. So pressure control hand wheel. So the pressure is applied, or it can be controlled by this B part. And then the C part is the terminal one. It is linear variable displacement transformer, so that allows the flow of liquid only in one direction. You can see that there are boxes which are mentioned at the bottom: stainless steel and stay-light valve mechanism. So this valve mechanism is very essential, which is shown by boxes with lines. In the center you can see boxes with lines. So this mechanism is responsible for non-returning of the slurry. That means the slurry, once it is entering inside this homogenizer, it will enter through a non-returning wall, and forcefully it is allowed to bombard on the surface, and this bombardment of microbial cells on the surfaces along with liquid, this force is responsible for the breaking of cells. So from this end, the microbial slurry, means the liquid containing microbial cells, are fed in, and they will enter into this valve mechanism, that is a non-return valve. So forcefully this slurry is fed into this uh, small non-returning valve, which will enter through a small orifice. So this is responsive. So bombardment of these microbial cells on the surface here and the valve mechanism, it will create the force, and that force will cause the lysis of cells, and then this slurry is allowed to pass through a small orifice as it is shown, which is connected to an outlet. So this is how this homogenizer it works for causing the lysis of cells to liberate the intracellular products. Let us see this in detail with the help of a video. As we can see here, the process of homogenization. So homogenization, it can be for the preparation of emulsions, or it can be useful for the preparation of slurry. So this is the diagram of high pressure homogenizer. So as you can see here, the microbial cells are getting bombarded onto the surface, and where they are going to be broken, so that there will be generation of pores or holes in microbial cells. So this is how the cells are broken, like this. So microbial cells are coming out and they are broken down. So they are bombarded onto the wall, and so that is how the homogenizer works. Now this is a mixer homogenizer, a different version, advanced version of homogenizer. So this is the assembly of that. So this is the device, how it works. So microbial cells are entering in the device, and this is how they are getting lysed. These types of machines can be also useful for the homogenization of different liquids also, like milk. This is how the whole system works. This is the whole assembly. You can see here the microbial cells are entering, and the broken cells are then fed back to the chamber, returning back to the chamber. This is another version. This is called as uh, uh, a combination of mixer homogenizer like this. Okay, then coming back to the methods, the next method is solid shear, that is cutting with solids. So this involves the pressure extrusion of frozen microorganisms at around minus 25 degrees Celsius through a small orifice. So the method is very simple. The microbial suspension is frozen at minus 25 degrees Celsius. So that leads to the generation of ice crystals, and this slurry is the frozen slurry of microorganisms is then fade into, or it is allowed to pass through a small orifice. So the disruption of the cell membrane and cell wall will result due to a combination of liquid shear through a narrow orifice and presence of ice crystals. So both liquid shear and ice crystals, that is the solid shear, are responsible for causing the lysis of cells, bringing the intracellular metabolites outside. Another method is agitation with abrasives. So abrasive is the material that is going to cause the damage to the cell wall and cell membrane. So this is a type of mechanical cell disruption in a disintegrator containing discs and beads. So this is the design of that mechanical cell disintegrator. We can see the design. So it is labeled by numbers 1 2 3 4 5. Let us see what are the components. So number 1, it is the, so this is the inlet for the microbial suspension. Number 2 is the manometer that monitors the flow rate of the passage of slurry. Then the third one is the rotating disc. So this device is just similar to the impeller, but only the position is horizontal. Then there is a fourth part, that is there is a slit for separation of glass beads from the suspension. So we can see that this horizontal uh, rotating disc, so it is rotating along with the glass beads. So glass bead will act as abrasive material. So glass beads will bombard on each other, and in between, if microbial cells are trapped, these microbial cells are going to be lysed. That is the strategy. So the fourth part is a slit. So once the disintegrator is run, uh, after the operation is stopped, then the slurry is recovered. The broken microbial cells are recovered. They are allowed to release from this slit. That is the fourth part. Then the fifth part, it is the outlet for the suspension containing lysed microbial cells. To regulate, to monitor temperature, there is inclusion of thermometer, that is the sixth part. Then the seventh component is, so as this is a rotating device along with the glass beads, this may generate a lot of heat, and if intracellular metabolite is heat sensitive, then it may leads to its destruction. So in order to protect these microbial metabolites which are sensitive to heat, the temperature is monitored and it is then controlled by the passage of cooling water through this seventh part. So that indicates the inlet and outlet for cooling water. Then other parts are mechanical, that is 8th, 9th and 10th. That includes bearings, then belt drive and the motor. So this is the design of this mechanical cell disintegrator. So the slurry is fed in from number 1, and this is how the device is rotating. You can see here, and the red circles you can see inside, these represents the glass beads. So slurry is coming inside, so rotation will cause the bombardment of cells onto the glass beads, and these glass beads will be responsible for the lysis of cells. Once this batch is run, then there will be concentrate on the fourth part here. After this, yes, you can see here that the slit is now open, that is the fourth part, and that will allow the passage of, you can see here red arrow, it will allow the passage of uh, slurry of broken microbial cells outside from the part number 5. This is how it operates. Then the next method is freezing thawing. We understand what is freezing. Freezing involves the exposure of microbial cell suspension to low temperature below 0 degrees Celsius, approximately in minus 20 degrees Celsius, and then return increasing its temperature, that is called as thawing. So freezing means bringing the temperature down, and thawing means bringing the temperature above 0 degrees Celsius. So this freezing and thawing, it is repeated several times. So that brings the lysis of the cell. So freezing and thawing of microbial cell paste will unavoidably cause ice crystals to form, and these ice crystals during thawing process, these ice crystals will lead to some subsequent disruption of cells. Then another method is ultrasonication. Ultrasonication, it involves the high frequency vibrations, that is 20 kilohertz, and that is generated through a device which is called as ultrasonicator. So it is generated at the tip of an ultrasonication probe. So this probe is just like a thermometer probe or a pH electrode probe, which is made up with stainless steel, and this is going to generate the very high frequency sound waves, and these ultrasonic sound waves are responsible for the creation of shearing conditions. It is responsible for the generation of vibrations which are intolerable. So if we are handling this equipment, then you need to keep the equipment in a box which is sonication proof. That means if these waves are so harmful that it may create damage to your ears also. So these ultrasonic waves generates vibration, and these vibrations are responsible for the creation of pores in, it causes the destabilization, it causes the destabilization of cell membrane, because cell membrane, it consists of phospholipid bilayer. So this fluid mosaic model that we have learned, so this phospholipid bilayer, it gets disturbed, it gets destabilized, and that leads to the formation of pores in cell membrane as well as in cell wall. So these shock waves thus produced are responsible for causing the disruption of cells. Ultrasonication, this is frequently this is one of the costly techniques, and this is generally useful for handling of smaller volumes of microbial slurry. So generally for research purpose, this kind of technique is used. This is the diagram. This is the probe that generates ultrasonic waves that creates vibrations, and suspended cells are then lyse to release the intracellular contents. Then there are several chemical methods. Only the drawback of chemical methods is that these chemicals cause the contamination of the product, and we need to take extra efforts for the further purification or the removal of these chemicals. Or these chemicals sometimes may be damaging. It may cause damage to the desirable fermentation product also. So in general, the chemical methods are not chosen. So if the product is relatively stable to all these chemical agents, then chemical methods can be used for causing the lysis of cells. A very common method is use of detergents. So as we know that detergents are the destabilizing agents which are responsible for the solubilization of lipids and oils, and cell membrane, of course, it consists of lipids, so easily it can be washed away and it leads to the generation of pores. Along with that, detergents are also responsible for causing the aggregation of proteins, so easily proteins can be aggregated, and that creates the destabilization of proteinic structure present in which may be present in cell wall. So detergents will damage the lipoproteins of microbial cell membrane. So for example, these detergents are quaternary ammonium compounds. Most frequently used is sodium didosyl sulfate, that is called as SDS, sodium lauryl sulfate, and triton X100. Then another method is osmotic shock. So in this case, uh, as we have learned about osmosis, so this is related to the movement of water molecules from high concentration solute zone to low concentration solute zone. So if you recall that if red blood cells, that is our red blood cells, if a drop of blood is added to distilled water, then what will happen, that distilled water is a hypotonic solution, means the concentration zone is very low, whereas red blood cell, inside the red blood cell, the concentration zone is very high because it contains multiple millions of molecules. So water will move from low concentration zone, that is from outside to inside. So in this case, the water will move inside, causing the lysis of red blood cells. So the same thing happens in case of microbial cells. So in this case, microbial cells are poured into a hypotonic solution, so that the movement of water inside will cause the bursting of cells. Then another method is treatment with alkali. So alkali treatment will bring the hydrolysis of microbial cell wall material. And finally, the last treatment, which is a costly technique because it needs the use of enzymes. So it is enzyme treatment. Enzymes are costly material. So generally for research purpose, when the cell lysis is required, enzyme treatment, it may be an affordable option, but not for the treatment of microbial slurry in big volumes. So these enzymes are lysozyme, cell wall degrading enzymes, lysozyme, which causes the lysis of degradation of cell wall of bacteria. Then enzyme extracts from leukocytes. So if you remember, we have learned in immunology that leukocyte, that means the white blood cells. White blood cells contains these lytic enzymes which are present in lysosomes. So this enzyme extract from leukocyte will be a good option for as a source of cell wall degrading enzymes. Then these enzymes can be recovered from streptomyces actinomyces type of bacteria. Then micromonospora, this is also a type of actinomycete. Penicillium, it is a type of mold. Trichoderma, it is a type of mold. And interestingly, snails will be a source of these cell wall degrading enzyme that may supply the enzymes like proteinase K, which is a type of broad spectrum protease. So that is all about the methods of lysis of cells.